In the back.
Didn't they find life in meteors that have hit the Earth?
They found something that looked like a bacterial cell, but as I showed before, just because it looks like a duck doesn't make it a duck. So they found something that looked like it, but there's been a lot of evidence put forward that you can actually form those structures that look like bacteria, and it has nothing to do with biology. It's actually geology that does that.
I don't think you mentioned anoxia as a factor. Is it in these organisms?
Repeat the question.
That's one. Is the lack of oxygen a factor in these organisms and how they respond?
Well, here's the interesting thing. Again, these things were built to survive the apocalypse. These halophiles can survive without oxygen. They have a form of photosynthesis that doesn't work for carbon fixation, but it actually works to create energy. It's the purple pigments on their membrane instead of the red ones. The purple ones will actually drive, so they can survive without oxygen. So a lot of these extremophiles are built to handle a range of different conditions. Yeah. Does that answer your question?
Back to question 1 over there, about finding the microbial deposits adjacent or among geological formations given a apparently known age of 250 million years. What are the causes for any suggestion of differential in the age between these two elements, if you will?
Well, from having discussions with geologists, because I'm coming from this from a biology point of view, they're saying that, you know, if you actually look at the evolution, evolution is the wrong word to use here, but how the mineral deposits are formed and how they can be changed over time by if there's water that moves into the area and then dries up again and that sort of thing. There's a possibility that even though the surrounding area is very, very old, that what you're looking at inside the fluid inclusion was actually trapped at a later date. So they can't be certain as actually when the bacteria were trapped inside. And I know that other people are working on ways to do that. And Mihaela in my lab actually works a lot with how do you identify remains of bacteria fossilized. So.
Do you come down with any assessment about that?
Okay, pure opinion on this is that 250 million years is a really long time for something to be alive. Because when they're trapped inside these salt crystals, they're essentially, it's almost like being in a coma. They're alive, it's not a spore. These cells are fully alive inside of these things. And there are studies going on right now trying to figure out exactly what they're doing when they're in that state. You know, what are they doing with their DNA damage? How do you repair 250 million years of DNA damage without the cell dying? How does the cell keep going that long? You know, what is it doing for energy generation? I think that's probably an overestimation. But again, I think the controversy is good because you can't just say, I don't believe you, you have to prove them wrong.
So you think there's been leakage in this older geology?
Yeah, I do.
Is methane ever involved? I asked because they've seen methane on Mars, and it's been a question whether it could be there without life.
Yeah, the methane, actually, the scientist who actually published one of the papers in the find, there's a European paper and an American one, is actually going to be coming to talk to us tomorrow about that. So we get to hear that. So I'm interested as well. But there's a signature of methane coming from Mars. And it seems like, given how much the signal that they're seeing, it seems like there should be a constantly replenishing source of methane. Like this wasn't a one-time phenomenon. But then the argument is this methane producing bacteria, which there definitely are archaea that are methane producing bacteria. And they work very, very well at high temperatures, depth. So are these bacteria responsible for it? Or are we looking at a geological process? And most people I've talked to are erring on the side of geology, especially the Mars methane findings. But again, you can think that all you want, but can you prove which one you have? So I think that the upcoming missions that they're planning for Mars, I mean, obviously the budgets for Mars missions right now is being really reevaluated because we just don't have the money we used to have. So it's one of those things where the scientists would love to go and find out the answer to that, but it's a matter of priorities at this point. There's a lot of people fighting for the same money for a mission to space.
If we turn the picture around a little bit and think of things that they you would think they would be tolerant to, but maybe toxic. Have you found any evidence of those sorts of scenarios?
Well,
instead of extremophiles, maybe they're supernomophiles.
yeah, it's, well, I mean, it gets interesting when you go into the extremophiles because they can't be taken out of their extreme environment. So, I mean, when I need to get DNA out of a halophilic cell, it's easy. I just drop them into water and then just pull the DNA right out because the cell's exploded. So we have things like that that happens all the time. What's really interesting with the archaea is if you ever try to kill them with antibiotics, and they've had very limited success trying to find anything that they're tolerant to. It either completely kills them with this ridiculously low amount, or they can take everything you've got and still keep coming. So it's been a really interesting process, and of course, I mean, using antibiotic resistance in genetics is one of the bread and butter techniques that we use to actually help study things. So not having antibiotics for these things is not very helpful for us. So a lot of people have worked on this, and it seems to be one or the other. They're either completely dead or completely alive with antibiotics.
More questions?
Marilyn.
Are the extremophiles known to be infected by viruses, phage particles?
Yes, yes, they are. There are phages and viruses for extremophiles. So I mean, you're never immune from these things.
Do you think that confers any, gives them their extremeness?
Well, there's sections of the genetic code that can be transferred from one organism to another, and viral transfer has been thought to be one of these things that can actually move pieces of genetic material from one organism to another, and if it has something in it that can be advantageous and keep them alive, then that can be a mechanism where that gets transferred from one organism to the next to the next. So these things are possible, but it's getting back to where did these things come from, which of these organisms was first in the end. But I think viral transfer is one of the things that perpetuates some mutation and gives some organisms some advantage.
One more.
So if I get the extremophile flu, I might be immune to high pressures?
And then you wouldn't even need the yellow submarine.
Okay, well, as you can tell, we are a department of extremophiles. We like extreme conditions, and Adrienne's an example of the quality of postdocs we attract from around the world. Let's thank her once again.